but what is being asked here is that, which is quite this question, this question came in the year 2020, and it is again asked in the section of botany, and it is a numerical aptitude based test question, and it carries 2 marks. So what it was asked that a double homozygous mutant develops green and wrinkle seeds when it was crossed with a true breeding plant having yellow and round seeds. All the F1 plants developed yellow and round seeds. After self fertilization of F1, the calculated percentage probability of plants with green and wrinkle seeds in the F2 population round off to 2 decimal places is. So if you have come across this kind of question, so this is a just a basic question from Mendel's dihybrid cross. So for understanding how to solve this question, now let us look into the different Mendel's laws that is being provided. So as you know that the Gregor Johann Mendel, he is the father of genetics. Okay. So when he started his experiments with the pea plants, so there were different means phenotypes that you should know that which is the dominant and which is the recessive phenotypes. So it will be actually easy for you to solve these kind of questions. So that's why I have given a table where you will know that what are the dominant phenotypes of the pea plants and what are the recessive phenotypes. So for flower color, the dominant phenotype is purple and the recessive phenotype is white. Seed color is dominant is yellow, recessive is green. Seed shape, it is round, the dominant is round, whereas the recessive is wrinkled. The seed, the pod color, it's the dominant is green color, whereas the recessive is the yellow color. Pod shape is inflated, dominant, and recessive is constricted. Flower position, if you consider the flower position, axial is the dominant and whereas the terminal is the recessive. And the plant height, tall and dwarf. Tall is the dominant phenotype and the dwarf is the recessive phenotype. So this, if you have the basic idea what are the dominant and the recessive phenotypes, that will be actually easy for you to solve these kind of questions. So in summary, what Mendel's principle states. So there are 3 Mendel's principles that we know from class 12, but I am not going into the details because we need to solve the question. So I am just going what is the summary of Mendel's laws. Okay, so the first law is the law of dominance. So the summary of law of dominance is some alleles are dominant and cover up the recessive alleles. So if you know that there will be the parental allele and then there will be this segregation that will happening, and in the first F1 generation, so there will be this heterozygous alleles will be forming, but the what is the phenotype that it will show, the phenotype will be showing the dominant phenotypes will always cover up the recessive allele. So if you have the alleles of both the tall and the dwarf, then the tall phenotype will be on you will be overcoming the dwarf phenotype and you will see the plants are tall. Okay, now this is the law of dominance. Now law of segregation. So what is law of segregation? An organism has 2 alleles for each gene, but they can only pass on one. So there are 2 alleles for each gene, but when they segregate for the F1 generation, when they will again come together, so when they segregate, there can be each gene, but they can only pass on one allele. Only one allele can be passed in the next generation. So this is the law of segregation. Then law of independent assortment, which means the genes found on separate chromosomes are inherited independently of each other. So in each chromosomes, if you this law of independent assortment is mostly discussed when you have 2 phenotypes. So genes found on 2 different chromosomes, for example, the genes for the height of the plant height of the pea plant and the flower color of the pea plant. So these genes were found on 2 separate chromosomes and are inherited independently of each other, but they are actually inherited independently of each other. So this law of independent assortment is very useful for solving the question that we have just discussed. Now let us look into more into the law of independent assortment. So this is the means a typical monohybrid cross, monohybrid Y, because there is only single phenotype which is involved here. That is this single gene which codes for the height, means which actually which actually codes for the height of the pea plant. So this TT is the um, those two alleles in two chromosomes, parallel chromosomes, and this small tt is the recessive alleles. Okay, now if you segregate them, T and small t will come from each means parental allele or phenotype. There will be each allele which will be coming according to the law of segregation, and then they will again in the F1 generation they will form a heterozygous parent. Okay, so this, if you cross this F1 generation, if you cross again with the means the heterozygous if you self fertilize, then you will get this Punnett square or this table. So where each t will be each allele will be representing in the horizontal and the vertical columns, and from there what are the possibilities. So if this t and t will give you the tall phenotype. This is the heterozygous genotypes, whereas the small t and the capital T are the homozygous genotypes. This capital T small t and capital T small t, all these three will give the tall phenotype because the dominant will overcome the recessive allele according to the law of dominance, whereas the small t, because both of the alleles here are recessive, it will only give you a dwarf phenotype. So if you now look into the phenotypic to the phenotypic ratio, that is tall to short, then you count, that is 1 2 3, 3 are tall and only 1 is the short, so the ratio will be 3 is to 1. Now the genotypic ratio here is 1 is to 2 is to 1 because two heterozygous are present, whereas these two are different genotypes, so that's why 1 is to 2 is to 1. So this is the typical ratios for a monohybrid cross. Now if we consider a dihybrid cross, where you are considering two genes coding for two different phenotypes, like for this Y is actually coding for the yellow color of your seeds, whereas this R is actually coding for the round or wrinkled. Okay, so this yellow and green is coded by this capital Y small y, and this round and wrinkled is given by this capital R and small r. So both of them are present. Now how they will actually segregate and they will form the F1 generation. So again, from each gene there will be each allele which will be coming according to the law of segregation. So Y capital I and capital R is coming from this parent, and small y and small r is coming from this parent. And in the F1 generation, you will again get a heterozygous phenotype, right? Heterozygous, you will get a heterozygous genotype. And this will be because this is the dominants are present. So this phenotype will be similar to the dominant phenotype that you have observed from the parent. Okay. Now, if you again self-fertilize, you make a cross with Y, capital Y, capital R, small r. In F1 generation or self-fertilize, then again you write all these alleles in the x and y direction, or in the vertical or the horizontal columns, and again create this Punnett square. Okay, so then you will be knowing that according to the phenotypes, just you need to remember that capital R codes for round, which is the dominant allele, and the capital Y codes for yellow, which is the dominant allele again, whereas small y and small r, these are the recessive alleles. Small y code for green, and the small r code for the wrinkle. So on the basis of that, you just see what is the genotype and phenotype, and you will find the ratios. So the easy way to do is that if you remember the phenotypic ratio of your monohybrid cross, that is 3 is to 1, which is very easy to remember. Now if you put 3 is to 1 multiplied by 3 is to 1 and you multiply like this, 1 multiplied by 1 is 1, 1 multiplied by 3 is 3, then again 3 1 multiplied, so 3, and then 3 3 then 9. So the ratio is 9 is to 3 is to 3 is to 1. So this is very easy how you can remember. Again, if you see the dihybrid cross, again if you write the monohybrid cross two times and multiply, then 1 is 1 into 1 is 1, 1 2 the 2, 1 1 the 1. So again if you 2 1 the 2, 2 to the 4, and 2 1 the 2. So again 1 1 the 1, 1 2 the 2, and 1 1 the 1. So this is the how you actually calculate the dihybrid cross. Now if you someone ask you to calculate the phenotypic ratio of a trihybrid cross, so you just need to multiply a dihybrid with the monohybrid cross, so you will get a trihybrid cross. So in this way you actually get the ratio. So this is a trick how if someone asks, this is a very basic, but still I wanted to tell you that. Now let us, since you understand this, this is similar to what has been asked in the question. So now let us go to the question again. So a double homozygous mutant develops green and wrinkle seeds. So there is a double homozygous mutant. So there is a homozygous mutant which develops green and wrinkle seeds. They develop the green and wrinkle cells. So green is also recessive. If you see the green color, and wrinkled is also recessive. So that's why this is a double homozygous mutant. This will develop green and wrinkle seeds. When it was crossed with a true breeding plant having yellow and round seeds. So yellow means yellow is the dominant allele and round seeds are the means your means round is also the dominant allele. All the F1 plants developed yellow and brown seeds. So all the F1 plants will develop yellow and brown seeds. If you see, so this is the scenario what is going on here. So if it is having the yellow and round seeds, so the homozygous mutant is actually crossed with this true parental generation, and all the F1 generation you are getting is a yellow round seeds. Okay, so that is being asked in the question here. Now, when it was round seeds, all the F1 plants developed yellow and round seeds. After self-fertilization of F1, the calculated percentage probability of plants with green and wrinkle seeds in the F2 population. So what they have asked is that they have self-fertilized this F1 generation or crossed it with it only. So YYRR is crossed by capital Y small y, capital R small r. And then what will be the scenario that you need to draw like this for solving this question. So I have drawn that. So again, I have taken the parents that is yellow round. Okay, so it is not given that it is the homozygous. So this is the parent which will give you capital Y capital R and this small y small r. If you cross them, you will get this heterozygous F1 generation, which is your male gametes of F1, and female gametes of F1 is also I have written just like in the Punnett square. Now I have just made the cross and written what will be the genotypes and what will be the phenotypes in the bottom of their genotypes. Okay, so now what they have asked that what is the probability of green wrinkle seeds. So the green wrinkle seeds, they have found asked us to calculate the percentage of green wrinkle seeds. So if you see that the green and wrinkled, so there is only one which is the green and wrinkle, that is both of them should be recessive. So the answer will be 1 and the total is 16. So the probability if you calculate, so that your observed the required quantity divided by total, that is 1 by 16, and the percentage if you want to calculate, that is 1 by 16 into 100%, that is giving you 6.25%. So this is the way how you can solve these questions. For your reference, you can follow these two courses of NPTEL. So the first course is Functional Genomics, and the course instructor here is Professor S. Ganesh of IIT Kanpur. And this second course that you can follow is Genetics and Genomics. Here the course instructor is Dr. Josh T. Puthur, and he is from Department of Botany, University of Calicut. Thank you.